The SMARCA2 missense mutation from lysine to arginine at amino acid position 951. The SMARCA2 gene is located on chromosome 9. Its wild-type function is to provide a template to create one subunit for SNF complexes. These complexes regulate gene expression through chromatin remodeling. Some examples of this remodeling could be forming euchromatin or heterochromatin.
The variant we research is a missense mutation. It has an autosomal dominant inheritance pattern. Instead of coding for lysine, position 951 codes for arginine. This mutation causes Nicolaides-Baraitser syndrome.
This syndrome is extremely rare, affecting less than 1 in every 1 million individuals. Its symptoms appear at a very young age, typically around as a newborn or infant, and it causes sparse hair, severe intellectual disabilities, brachydactyly, among other symptoms.
In the wild-type variant, position 951 codes for lysine, which contains an amine group. Lysine is a positively charged amino acid because of this amine group.
And the mutation is a missense mutation, causing arginine to be coded for instead of lysine. Arginine is similar to lysine in that it has a positively charged side chain. However, unlike lysine, arginine has a guanidino group, which contains 2 additional nitrogens. This allows for additional hydrogen bonding, leading the protein to fold differently.
We decided to investigate the SMARCA2 gene and initially researched it on simple ClinVar. All of the variants listed in simple ClinVar were recorded as a pie chart and scatter plot in Microsoft Excel. We chose a specific mutation from the scatter plot and sequenced it on polyphen-2 to observe its prediction model and see how likely damaging the variant would be. A FASTA sequence was obtained from Uniprot to render the 3D model of the wild-type and mutant protein on Yosera.
And figure 3 shows a scatter plot showing all the recorded missense mutations for the SMARCA2 gene at each position and CAD score using data from simple ClinVar. The variant we researched is uncertain, and it has an amino acid position of 951 and CAD score of 4.39.
The prediction model from polyphen-2 predicted that this mutation is probably damaging, with a score of 0.989.
Figure 5 shows the rendering of the wild-type protein, and figure 6 shows the rendering of the mutant protein.
Figure 7 shows a 3D model of an overlay of the wild-type and mutant that was rendered on Yosera.
And figure 8 shows a close-up view of the wild-type amino acid lysine overlaid with a mutant amino acid.
We chose our specific mutation in position 951 because it caused Nicolaides-Baraitser syndrome and had an uncertain clinical significance. The specificity and sensitivity score in figure 4 was 0.989, meaning there's a 98.9% chance that the mutation is probably damaging.
The 3D proteins modeled in figure 5, 6, and 7 show how similar the wild-type and mutated proteins are. This is further demonstrated by figure 9, which shows that the wild-type and mutant proteins have a calculated 99.93% similarity score. However, the differences in the protein structures responsible for the damage can be seen in figure 8, showing the single amino acid change from lysine to arginine. The fact that this single amino acid change caused such a high probability for damage means that the protein's function is very dependent on its shape.
The missense mutation from lysine to arginine at position 951 has a high probability of causing damage. Since there are more nitrogens on arginine than lysine, there are more possible hydrogen bond interactions that can lead to a misfolded or misshapen protein.
Research on this variant should be further explored due to the high probability of the mutation in the SMARCA2 gene being damaging. Furthermore, the disease Nicolaides-Baraitser is extremely rare and very overlooked in the research and healthcare community, giving it more need to be researched.
These are references that we got our data from. And a special thanks to the developers at Yosera for allowing this presentation to be possible.